Each year, Microsoft Research hosts hundreds of influential speakers from around the world, including leading scientists, renowned experts in technology, book authors, and leading academics, and makes videos of these lectures freely available.
Next, we're going to have a talk on protein foldings, RNA folding and structures. I apologize. By Professor Robin Guttell and David Gardner. Professor Guttell is from University of Texas at Austin, and he's been working on cell and molecular biology, particularly RNA folding, and applying computational methods to that for the past 25 years. And joining him on the talk is going to be a graduate student of his, David Gardner, who's got a master's in applied mathematics and currently is working with Robin on in the Institute of Cell and Molecular Biology. So.
Okay, this is a project that I started as a graduate student, and it's one of these types of projects that you just can't let go of, and so I anticipate to continue to work on it probably until either I solve the problem, of which we've actually made great progress, and that's what we'll talk about today, or I retire. It would actually be nice to solve it first so that I could retire sooner, but so the title is Improvements in the Determination of the Fundamental Principles of RNA Structure and Prediction of RNA Secondary and Tertiary Structure. And in one sense, you might think that the problem is simple, and that is that you take this sequence, in this case it's tRNA, which has 76 nucleotides, and you want to fold it up into what's called a secondary structure, and then fold that up into a 3D structure. And in general, a secondary structure is composed of a helix that has 6 different types of base pairs: GC, CG, GU, UG, UA, and AU. So 6 of the 16 possible pairings are formed and are arranged to form a helix. So here's a quick outline of the talk. So I'll talk first, and then David Gardner. I'll talk about the importance of RNA. It's considered one of the grand challenges in biology, the RNA folding problem. I'll briefly talk about the description of the current concepts used to predict an RNA secondary structure, the physical dynamic aspect, and the traditional energy-based methods, and then the current meaning the way that other people do it, not the way that we do it, the accuracy. And then I want to briefly talk about comparative analysis and how we use multiple dimensions of information, about 5 or 6 different dimensions of information that we put into a SQL server. And then at that stage, I will let David Gardner, who is the computer scientist, talk about more of the computational aspects as well as the successes that we're now having on this project. Okay, briefly, RNA, for those of you that haven't been following in the last few years, but there's a major paradigm shift. Everyone used to think that proteins is where all the action was, and now they're finding that while proteins are still important, that RNA is far more important than people ever anticipated. In the human genome, there's somewhere between 20 and 40,000 genes. This is about the same number of genes you'd get in a C. elegans, a small worm, in Drosophila. It was always thought that humans would have at least 100,000 genes. That's not apparently what they're finding. So only about 1.5% of the entire genome for a human is actually coding for protein. And so that is very strange. But what became more unusual was that they noticed that over 90% of the genome is actually transcribed into RNA. So you have some people that think that actually what it's doing is you're just making lots of RNA and the cell's just breaking it down and the cell's just not very efficient. And then there's other people, of which I'm in this camp, that feel that no, this RNA that's being transcribed is in some way that we don't completely understand yet, very important in how the cell functions. And the second point is, in one sense, I'll just mention it quickly, that it was thought that the 3 major RNAs, ribosomal RNA, tRNA, and messenger RNA, were essentially the only RNAs that are in the cell that are forming, or that have a function and are important. My PhD and most of my work has been on the ribosomal RNA. And then, as I was alluding to, this perspective, this paradigm is shifting, that tRNA, messenger RNA, and ribosomal are the only important RNAs. And there's some studies that are now beginning to show that the complexity of an organism actually scales with the amount of RNA in that cell. So organisms that are not as complex have a smaller genome and or transcribe less of its genome into RNA. So all of this is brand new. It's coming out. Whoops. And just within the last few years, they've given several Nobel Prizes for people that work in RNA, especially this year. And I'll talk about one of them in a few slides. So in physiology and medicine, one of the Nobel Prizes was for a group of people that worked on a complex that involved RNA. And the second group worked on the ribosome, of which the ribosomal RNA is a major component. Okay, so our goal is to take a RNA sequence and fold it up into a secondary structure. So here's the tRNA secondary structure. And what's here is the tRNA sequence in both the X and the Y direction. And below the diagonal are all the potential helices. A helix, again, is composed of AUGC base pairs that are consecutive and anti-parallel. And so you see you have a fair number of those. And what's above the diagonal are those that are actually in the real tRNA structure. So our goal is to essentially determine how to predict these 4 helices out of all of these. And I'll jump down here. Here's tRNA. It's about 76 nucleotides long. There's about 37 potential helices for this tRNA. These can be arranged in many, many, many different ways. The number is 2.5 times 10 to the 19th. And there's only, as this shows up here, there's only 4 helices that are, 4 of these 37 are real. If you look at larger molecules, like the ones that I studied, starting as a graduate student, 16S is like 1,500 nucleotides, 23S is just under 3,000. Number of potential helices, about 15,000 for 16S, over 50,000 for 23S. The number of possible ways that these helices can be arranged into a secondary structure are these astronomically large numbers. And so if I knew how difficult this was when I started graduate school, and my PhD was to actually solve the secondary structure for 16S, I think I probably would have changed topics. I'm glad I didn't. Okay, so here's how people, so how do you predict a secondary structure? The goal all along has been to find the most stable structure. So here's an example where you have a helix that's 10 base pairs long, and the stability of that helix is minus 26.1. So it's the same stability for both of these. But this loop is considered to be destabilizing, so it's got a positive value. Minus value means that it's more stable. And then the hairpin is the entire unit. And so what you see here is that when you have a loop of just 10 nucleotides, its stability is minus 21, which is just slightly more stable than this hairpin that has a hairpin loop of 200, meaning that this helix right here, just by itself, without forming any other structure, is almost as stable as, say, this helix right here. So it had occurred to me, and it's occurred to other people too, but that regions of the sequence that are very close together are much more likely to interact with one another. And in one sense, that's very simple. Florey got the Nobel Prize, I think, in 60 or so, for studying polymers and just showing the regions that are close together on a polymer are more likely to interact with one another. And so we now look at it as, here's this tRNA, and let's just look at this small window that's along the diagonal, which means that all of these helices in this range right here are forming between a sequence or two sequence at the same sequence, but that they're close together. And the heavy red show those that are actually in the comparative structure. And so stage one of our folding model is that you form these local short-range helices first. And then once you form these three, which are shown here boxed in red, and especially once you form these two outer ones, these two regions are now very close together. So we had called simple distance just the distance along the sequence, but once you form these first set of helices, you then we now call it a conditional distance, since these two regions are now close together. This helix here, which is this one in green, is actually very, very close to the ends of these. So again, it's this issue that once you bring regions close together, they are more likely to form a structure. And so what this implies is that the order of the folding is extremely important. You have to fold the short range first, and then that brings together other helices that were far apart in simple distance, but their conditional distance now is much, much shorter or smaller. Okay, so this is how people currently look at this, currently determine the stability of a helix. So for each two consecutive base pairs, it's given a value. So these two base pairs here get a value of minus 3.4, and the next two get a value of minus 2.4, and you essentially add all of these up, and that gives you the stability of this entire helix, which is minus 13.4. And you can do that for the second helix here and for the third. And so the current folding algorithms essentially look at a helix and say, look at a sequence, determine all these helices and say that's where you get stability. And then they look at all these unpaired regions, and I say that unpaired in the secondary structure, and they say that they are destabilizing. So each one of these regions that don't have any base pairs in the secondary structure are destabilizing, and that is part 2 of the folding model, which is that we don't know how to determine the energetics correctly. So again, part 1 is, going back, is that the current method essentially assigns an energy value for every helix at the beginning. So again, it would give this helix a energy value that's just slightly less stable than this helix right here. And we're saying no, that these helices actually form in a specific order, short range first, bringing together regions that were far apart into proximity. Second part of our folding model says that the current methods for actually determining the energetics, or our knowledge of the energetics of RNA structure, is overly simplistic. This is how this structure right here actually looks in 3D, which is that this is, instead of these being unpaired, they're actually base paired. It's not a standard type of base pair, but they're paired. We currently don't know the actual energetics of this. I'll skip this. This is how this region looks in 3D space. The main idea is that these loops in the secondary structure are really not loops. They're essentially forming lots of structure with one another. And what they're doing is called base stacking, that the bases in these atoms, in these nucleotides, stack onto one another, and that's where you get most of your stability. And it's a little lighter than I anticipated, but this is the secondary structure for a 16S ribosomal RNA from this organism, Thermus, thermophilus, and this is the crystal structure or the 3D structure that that was actually determined in the year 2001 or 2. And the main take-home lessons is that in the secondary structure, there's like 936 base pairs and about 62% and about almost 600 unpaired nucleotides. But in the real crystal structure, about 79% of the nucleotides are base paired and about 319 are not paired. And so when you use the standard methods right now to predict secondary structure for 16S, 23S, and a few other molecules, for 16S, the average, 10, 20, 30, 40, is about 40%. And with one standard deviation, it goes from like 50% down to 30%. And for some people, that's good enough. But of course, for people like me and many other people, that's not acceptable. And I'll just say very briefly, it was noted that for helices that are in very close proximity, and for helices that have a very short simple distance, like 2 to 100, the accuracy is actually much higher with these methods. And as the simple distance increases larger and larger, the accuracy actually decreases. As the simple distance increases, the accuracy decreases significantly. Okay, so how do we get the energetics for all of these structural elements that cannot be easily determined from experiments? Because experiments cannot determine the energetics for all of these different types of structural motifs, elements that have not even been identified yet. So, as a graduate student, I started using this method called comparative analysis. And long story short is that different sequences will fold into the same secondary and tertiary structure. And so for tRNA, these 3 different sequences that have like 40-45% sequence identity with one another all form essentially the identical secondary and tertiary structure. So I spent many years analyzing these, and we now have over 100,000 different, essentially comparatively solved 16S, 23S tRNA molecules. You should ask, how accurate is this? How accurate are these comparative structure models? So here's a comparative structure of 16S, 23S. Here's the crystal structure of the 30S, the front and back sides. And the answer is that about 97-98% of the base pairs that were predicted with comparative analysis are in the crystal structure. But that's the secondary structure base pairs. With comparative, we cannot predict the tertiary. And I'll just note that last Wednesday, they actually gave the Nobel Prize to 3 people that solved the crystal structure for the 30S and 50S. And it's unfortunate, my PhD advisor was one of those people that solved it, but more unfortunately, there were 5 people that solved it, and they only give 3 Nobel Prizes, and his name wasn't picked out of the hat, unfortunately. But, okay, so I'd worked with Carl Woese, and he had made this very profound statement in 1983. He said that the comparative approach indicates far more than the mere existence of a secondary structural element. It ultimately provides the detailed rules for constructing the functional form of each helix. Such rules are a transformation of the detailed physical relationships of a helix and perhaps even a reflection of the detailed energetics as well. There might envision a future time when comparative sequencing provides energetic measurements too subtle for physical chemical measurements to determine. To me, this is just an absolutely amazing statement made in 1983, because this is exactly what we're doing, and this is what David's going to talk about. And I think this is almost my last slide. We have multiple dimensions of information. Here it's a diagram in the upper left that's meant to show a alignment of sequences. We have alignments that have several 100,000 sequences in them. So it's a huge amount of information. All of these organisms are mapped to an organism that's on the phylogenetic tree of life. We've got the tree, we have the crystal structures of many different RNAs. We've got we essentially have a secondary structure for every sequence. There's different structure motifs, as shown here and here. And we've essentially put all of this information into our SQL server, and that was done by Stuart Ozer when he was working in Jim Gray's lab. And so we've done that, and yes, okay, so there's Jim Gray, top and bottom. Here's Stuart. And because I'm running over, I'm not going to read through this. Suffice it to say that Stuart essentially helped us organize the data, and David is now analyzing the data. And so I'm going to stop now and let David talk.
So to make better use of these multiple levels of multiple dimensions of data, we created the RNA Comparative Analysis database, which combines sequence data, sequence metadata, evolutionary data, and structural relationships. So just to briefly restate the problem, we're trying to predict right now the secondary structure using only the primary sequence. And so most methods right now try to solve it with just simple free-energy minimization, which basically looks for the overall structure that has the lowest free energy value. And so how well do these do? For smaller molecules, they actually end up doing not too bad. But for large molecules, they have big problems, especially like here for the eukaryotic, which this is just how many base pairs are they able to predict. And so for like the 16S eukaryotic, it's only like 34%. So so on the left side, what you have here is a bunch of sequences, and this is the overall accuracy. And down here is the accuracy for base pairs that have a simple distance less than 10. And as you can see, that it's very, very important to get the simple distance, the short range predictions correct. If you don't, it just basically cascades and you get terrible scores. And this here over on the right is just 4 specific sequences in greater detail. And so this is just the accuracy based upon simple distance. And so if you see here, when they do pretty well in the short distance, they actually have a chance of doing pretty good overall. But when they score poorly on the short distance, basically they cannot recover, and it just leads to very low scores. So our folding model is kind of a paradigm shift. It incorporates 3 basic ideas. Is that 1, distance is an important factor, so that you only search for helices that have short, simple distances. The second one is using comparative analysis to improve the energetics. The third thing is kinetics, that there is a direction to the folding pathway, that there is competition, and that this should be taken into account by the folding model. And so to detail the distance contribution, we did some analysis where we took basically we went through a molecule and we determined all the possible helices within the molecule based upon energy and based upon distance. And that when you're, first thing is that when it's more stable, it's more likely to form in the actual comparative structure. The second thing is that when it has short, simple distance, it's more likely to form as well. So the idea is that you look only in short range. And so this is kind of, so the question is, how does it hold for the next iteration? In other words, if this is formed, this helix is formed, and this helix is formed, does this distance constraint still hold? And so we come up with the concept of conditional distance. And so what we did was we assumed that all the hairpins in a molecule have nucleated and formed, and does its distance constraint still hold? And it still does. That as a short, simple distance gets smaller, the prediction accuracy goes up. But so distance does improve the prediction accuracy, but for most of the helices, they're actually pretty unstable. So this in and of itself is not going to be enough. You need to have better energetic parameters. And so one of the things we did, we've already published this paper, is what we did was we basically generated our own statistical potentials and replaced and took mFold's energies and replaced them with ours. We generated them with this equation, and this is how well we did. We improved. We took statistical analysis of 4 different types of molecules, combined them all to generate some statistical potentials, and when we used them, we improved the accuracy fairly significantly for the tRNAs and the 5S rRNA. The 16S rRNA didn't improve quite as much, but they still did see some improvement. And so this here is just a diagram of all the different types of, well, not all, but a lot of the structural motifs that we can generate energies for that are not currently handled by most free-energy minimization programs. And one example of that is this type of structure right here, this cap. This is not a regular base pair. It's not a base pair in the comparative model, but when you take this into account, when you have a GC at the end of the helix, and it's got an A and a G here, you have, this is the original results, and down here you can see that it's significantly improved the prediction accuracy by just including this simple type of structural motif. And so our goal is to generate a better statistical potential than what is current. Actually, they don't. The mFold and most methods don't even have an energy. They don't have an energy value for this. Another structural motif that we believe we can come up with much better energy values for, and I think this is probably going to be the most important one, are hairpin loops, nucleation points. And so what we did was we went through a sequence, every sequence in our alignment, and we looked for hairpin loops. We looked for all hairpin loops from length 3 to length 15 that are surrounded by 2 or more base pairs. We determined if it was in the comparative structure or if it was just a potential helix, a potential hairpin loop. And so down here, if you have a tetraloop, which is a hairpin loop with 4 nucleotides, like this, GAA, and if the closing base pair makes a big difference on whether or not is highly predicted. And so what you see in here is just this range right here. So you have a lot of hairpin loops, potential hairpin loops down here that never form in the comparative structure. So there are 2 take-home messages on this. is that there are some that form quite a bit, quite often, and there are some that are actually never form. And so our future work is we've taken we've started to implement all of these concepts into a folding model. We need a folding algorithm. We've gotten pretty good results already, but we need to finish the implementation and test it and refine it. And then that will lead into a tertiary structure prediction. And that is all.
Any questions? I've got one. How did you use SQL Server in your, do you actually implement any?
All of the analysis has been done in SQL Server. So we used to do all the data analysis, like this data analysis and this data analysis is all done in SQL Server. So all the sequences are in SQL Server. All the structure information is done in there. And so we just work completely in the SQL Server.
And how much of those tables and schemas are actually usable beyond RNA folding to other?
Oh, it's usable. I mean, our goal is to do curation of alignments, and so we'll be able to do that type of stuff too. Lots of different applications.
So, can you talk about how this, since you're going from primary sequence for folding, how this rolls up or compares or works hand in hand with protein folding from primary sequence? I'm sorry. I was just wondering. So, protein folding from primary sequence is also a thing that people are looking at. I was wondering if you could talk about the comparisons or where this overlaps, where it doesn't overlap.
So the biggest thing is that RNA folds has a fairly consistent secondary structure, has very consistent rules for what you see in the secondary structure, this here. Proteins don't have that anywhere like that. I mean, they have like beta sheets, and so for RNA, it's much easier. I mean, it's much more feasible to do it in 2 steps. We predict the secondary structure, and then you predict the tertiary structure. I don't think that you can really do this type of approach with proteins. You have to kind of...
Well, no, I would actually disagree in the sense that... is that one would definitely have to change the schema, but that's very doable. And one would have to change the type of information that you put in it, because proteins, you have beta sheets, you've got alpha helices, and what David was saying is you don't have the specific rules to form a beta sheet and an alpha helix like you do to form a secondary structure helix. But I'm curious, now that this approach is working so well, and I'll say something that David didn't mention because we haven't published it yet, so we didn't want to make a slide of it. Well, okay, first let me say that, is that when David has actually already developed a folding algorithm that essentially uses the distance approach, the simple and then the conditional, and then uses structural statistics to create pseudo energies, or some people would call them statistical potentials. And for some molecules, he can predict, his predictions are nearly 100%. So we're essentially using the information from our comparative data sets to make predictions that are extremely accurate. Back to the protein question, I'm very curious if one were to collect the right type of information from proteins and use the same general concept. The distance concept applies to proteins. So essentially what you want to do is to drive energetics from proteins using comparative analysis. So I can't tell you the details of how to do it, but I actually believe that using our general approach that we can improve the prediction of proteins.
Questions?
Any other questions?
Thanks to all the speakers in this session. Let's get started with